Yes.
So what's your underlying model of recombination and the recombination rate? Are those adjustable? Or is it all based on cell organ?
It's based on the genetic map, yeah. So it's not hugely adjustable. It could be tweaked, but it's, yeah. If you want to generate whole genome data, so I guess I would recommend in terms of using sim.mphos, I probably wouldn't recommend generating more than about 30 to 50,000 markers. I've done it. It takes a lot of memory and it takes a decent amount of time, so you need a relatively powerful computer. What is a better option is probably alpha MP-Sim, which is something I worked on with John Hickey at the Roslin Institute, and it's written in a combination of Fortran and R. It essentially generates a larger population and then will pick out founders for you based on certain criteria. So if it'll generate phenotypes for everything in the population, you can pick out the top performing lines as your founders if you want. You can pick out ones based on genetic diversity if you want. There's a lot of different options. It'll select the founders and then it'll just send them through a pedigree in the same way map does, but it's written to be faster and more memory efficient because it's really aimed at being used for whole genome data. Very large-scale simulations. And so that was published in Bioinformatics in 2014, and it's available on John's website. In terms of estimating power, you need to quickly. You're going to be generating lots of data. Depending on what you want to do, you can keep a large percentage of your observed data. You could also keep basically everything except your phenotype if you want to, but it depends on how generalizable your results need to be. So for example, if you keep your map, your founder genotypes, your progeny genotypes, the power analysis you do is not going to be applicable to any other population. If you say, well, okay, I want to know what the power is for a general four-parent design with this, this map, so basically localizing it to some organism or some sort of marker spacing with varying founder genotypes, varying progeny genotypes, or even with the same founder genotypes but different progeny genotypes, it's a bit more generalizable. And so that will affect your purpose is going to affect how you set up the simulation. Essentially, you're just analyzing each data set as you would the observed data. So you'll generate some QTL and you'll see how often you can pick them up. Generating individual QTL on a chromosome is the simplest way to do it, but it's not going to give you as good an idea of how your real data will perform as if you generate more complex scenarios. So it's a balance between how much time do you have to explore lots of different options versus, you know, realistic are these scenarios going to be. Polikover in the Arabidopsis paper has quite a bit of details on the procedure for how they estimate power and look at the precision of their QTL support intervals. So that's a good place to start to look for how to set up these types of simulations. And one of the things we use simulations for a lot is looking at recombination. So that's why I combined these two. Essentially, in order to count recombination events, we need to use the founder probabilities that we've generated. And we need to look at where we're switching between different founders. Now, one of the issues is that in the transition regions, we don't have as much certainty about which founder we're actually observing. So we tend to have lower probabilities for multiple founders when we're about to switch from, say, founder 2 to founder 3, simply because there's a bit of uncertainty. It's not nearly as certain as it is when we know it's founder 2 and we know it's founder 3. But we can estimate at the very least the number of recombination events pretty confidently, the number of times we're doing these switches. And this bit of code here is looking at basically that EST-FND matrix, that list of matrices that I mentioned before. And for each row, it's just looking at the number of times you're switching between different parents. So it's saying, okay, if we take the difference between the values, when is it not 0? So you're not going from 2 to 2, you're going from 2 to 3. And then if we take the mean of those, that tells you the number of recombinations, the average number of recombination events for your population. The problem is if you have errors in the map, it can cause additional recombination events. So this was based on the map that we generated. So this DAT final is the map that we generated. The other one, which I'm calculating here, is based on the simulated original map. And you can see that the average number of recombination events is a little bit lower. Okay? So something to keep in mind. It's not a huge difference, but you will tend to, for the most part, if you're constructing a map, it's not going to be perfect. You're probably slightly overestimating the number of recombination events. There's a good chance of it. That was based on basically just saying, okay, if we walk along the probabilities, which allele has the highest probability? So that's how we're determining our haplotype mosaics. There's another script in the directory called nrec.r, which is based on a slightly more sophisticated algorithm, a forward-backwards algorithm, which allows for a penalty for switching. And you can see that depending on what the penalty is, you actually get different values for that average number of recombination events. So if you have no penalty, you get 13.8, which is actually a little bit higher than what we had before. If you increase the penalty to 2, you get 10.9, which is a little bit lower. I'm still playing with this at the moment, honestly, to determine what the best penalty is. I'm not sure there's an obvious answer. So it's up to you. If you have no penalty, if you have errors in your map or so on, again, you will tend to inflate those estimates. So you have to figure out something which works well. And basically, this is what underlies simulating the recombination events, plots like this. So you're generating lots of data sets, you're counting the mean number of recombination events, and then you're doing a histogram over all of your data sets. So I didn't put it in the exercises, but if you want to, you could see if you could generate a data set, generate a plot like that. So this is the biparental for red, 4-parent for blue, and black, 8-parent for black histograms of the expected number of recombination events. It's a good exercise to go through for your map and for your population to see what you're expecting to see and how your reality compares with that. Now, you can also count per chromosome. This is the results for the estimated map with a penalty of 1 in this case. You can see that, I mean, you would pretty much expect if your chromosomes are the same length, you're going to get very similar numbers of recombination events. And that was what we saw. Chromosome 2, they seem to be a little bit higher, but actually in the map, if you remember, chromosome 2 ended up with a slightly longer estimated map length. So that's not too surprising. It may be a good check to see if there are any chromosomes where you have a very large number of recombination events. It's possible there's a recombination hotspot on that chromosome, but it's also possible you have some errors there. And you can also, once you estimate the number of recombination events, rather than take the mean, you can actually take those numbers for each line and do QTL mapping with that. So we've done this. Richard's done it with the Arabidopsis population. And although if you do this for the simulated data, you're not going to come up with anything. In real data, you often do see some interesting results. And it's possible to do this because you have the high resolution to detect these crossovers and you have a large number of lines, of inbred lines in these populations. So, any questions about recombination or simulation? Okay. All right, so as far as visualization goes, there's a lot of additional libraries. Like MPMap has some built-in plotting functions, but there are a lot of additional libraries that can be used that I personally try and use ggplot2. It's a bit complicated, but it's easier than Lattice. RCircos is nice for generating circular plots, and I'll show you an example of that. Heat Plus is what I use for heat maps, which are not interactive, and there's also one called LD Heat Map. R has lots of different packages, definitely. Too many, some might say, but the good thing is you can do just about anything you want in terms of visualization, and you can produce publication quality graphics if you need to. So I mentioned MPMap Interactive to you before. I'm not sure why there's a slash there, because it shouldn't be. But if you are interested in it, look on GitHub under MPMap Interactive. It's written by someone called Rowe and Shaw. That's how you'll know if you have the right one. And there's instructions there on how to install it. So at the very least, it works on Macs and Windows. In theory, it works on Linux as well, although I haven't tested it there. And that's what allows you to group markers, combine groups, remove markers, flip them, reorder chunks. And although it currently only works for MP cross objects, if someone's really interested in using it for more general crosses, the main thing you have to do is to calculate your recombination fraction matrix and then input it into the structure that's being created. So this is a question that came up, so maybe this is a good time to address it. How much of MP map can be used for other types of crosses than multi-parental? The answer is the only two parts which really rely on it being a multi-parental cross and which will produce incorrect results if you use something else are the estimation of recombination fractions and the estimation of probabilities. Both of those depend on the design of the population, okay? If you were to estimate the recombination fractions externally, so for example in RQTL for a biparental cross, or you were to estimate the founder probabilities externally, so using calc genoprob in RQTL or using HAPI or something like that, it's relatively easy to import things into MPMAP and use the functions which exist there. So most of them, they all operate on the structure of an MP cross object. So if we go back, we think about, well, we have MP cross, we have finals, we have founders, and then, you know, this ID, FID, pedigree. Okay. If you have a biparental cross that you want to pull into MP map and do some things with it, that you want to use in MP map interactive, actually I haven't tried it with MP map interactive, but I can play with that and let you know. This, you just need some sort of dummy matrix. It won't be used if you're pulling in the additional components, which are the recombination fractions and or the probabilities. So constructing a map, what you need to do is estimate these recombination fractions and note that this consists of three different components, two of which are really used, which is the theta, the actual estimates of your combination fractions between all of your markers, and the LOD score between all your markers. And these are both things that you can get out of s.rf in RQTL. If you're doing these probabilities, it takes a little bit more work because the format from calc genoprob is a little bit different than in MP map. But if you want to learn how to convert it, then you can look at the code for MP-prob and where it calls program equals QTL. Because this is essentially what we do. We take an MP cross object, we export it to RQTL, calculate the genotype probabilities there, and then we bring them back in. So if you want to do it for biparental cross, it would take exactly the same machinery. Basically, you would feed your cross into RQTL, you would then calculate your genotype probabilities and then run the code in that little section, essentially. Once you have these two, if you have these two in your MP cross object, once this is in, it's your MP prob, basically, then the functions aren't going to use the pedigree anymore. So it doesn't matter what's in there. This FID, this can be set to 1 to 2. It's not really important. And the same for the ID. None of those are really going to be used. In fact, when you're constructing the map, the main thing that's going to be used is this object. And that's why you can get around doing everything. So once you've computed that, you can then use all the functions. And it's just going to operate on that. It's going to reorder things. It's going to base it all on that. And then you'll get a map out at the end. Does that make sense, or do you want me to try and generate a data set in RQTL and convert it to an MP cross? Maybe, no? Okay, we'll leave that for later if we have time then. As far as built-in functions with MPMap go, there are plot functions for most of the objects. So there's plot.mpcross, plot.mpprob, plot.mpqtl. The dot there means it's an S3 function. So if you have an object of a certain type, it knows automatically which one to call. You just plot on it, and it will automatically do that. So plot, when called on an mpcross object, will show you the linkage map. It'll show you a histogram of the phenotypes, and it'll show you the recombination fraction heat map. If computed, plot called on mpprob, probably already mentioned, shows you the percent of the chromosome inherited from each founder, shows you haplotype mosaics for each chromosome, which can be subset to a certain number of lines, and it shows you the founder inheritance across the genome, which gives you an idea of segregation distortion. And finally, plot called on an mpqtl object shows you the QTL profile and the support interval. So that's the example for mpcross. You can see the linkage map, and you can see the histogram. If you want to plot it individually, there's a function called plot_link. Plot_link.map, and here you do have to write out the whole name. With that, there's an argument for setting the marker names to false. If you don't include that, typically it produces a huge list of text, which is not very useful. So that, without the marker names, obviously. Plot mpprob, just to show you some examples. This one is the first plot that comes up. It shows you the founder percentage by chromosome, and the white bit is the part that we can't identify. So if your map is not hugely dense, or if you have founders which are very similar to each other, then you may not be able to tell them apart, and you'll get large bits of white. Otherwise, you'd hope that those bands are roughly even. So you're hoping you should get roughly equivalent amounts of each chromosome from each founder. For some reason, this is very pixelated here, but this is what the haplotype mosaic plots typically look like. So this is just for one chromosome. You have the lines going down on the bottom here, the columns, and then you have all the markers along the side. And essentially, you're seeing the blocks which are inherited from each of the different founders. So using the same color scheme as in the first part.
Like most of the data, missing data is right where they switch.
Yes, that's typically true. So yes, if in those regions you tend to be much less certain about which parent it is, and so I think the cutoff for calling a haplotype is something like 0.7 for the probabilities. You can lower it to 0.51, which means that basically everything will get called, but sometimes it's good to know that's an uncertain region. And yeah, it keeps in mind that you don't actually know exactly where the crossover occurred. It was somewhere in there. And then that's the last plot that's generated. So as I said, you can use this to diagnose segregation distortion. For a four-way cross, you expect to see everything kind of hovering around 0.25. For an eight-way, it should be around 0.125. And for our eight-way, for example, in our four-way, on one of the chromosomes, it's basically double nominal level should be, which is because that bit of the chromosome has an introgression which has meiotic drive. One thing you will notice, though, is that even here, this is simulated data. There's nothing that should be causing segregation distortion. You still get bits which are deviating from 0.25. So here, on chromosome 3, the purple one is almost completely above 0.25. So there is random noise, obviously. So don't be too worried about segregation distortion in that case. In terms of comparing maps and validation, I mentioned the map comp function, which I won't go through all of this before because I talked about it. The summary function basically shows what I showed before, the correlations, identifying duplicated markers, and so on. And the plot function does a scatter plot of the marker positions for the two maps. Yeah, so that's all what that produces. And then in terms of interactions, I put in another function, an R script called circular intx, I think, .r, which generates this kind of plot, basically. So if you've computed interactions between all of your markers, and you set some sort of p-value threshold, and you just want to show the most significant ones, what it'll do is that it'll go around it'll put all of the chromosomes around a circle in differing colors, and then it'll put lines to show where the interactions are occurring between different markers. So you can see they tend to be clumped. You tend to have a group of markers on 2 different chromosomes, which tend to show interactions. And in this case, because wheat is hexaploid, I actually made it so that within groups, homology groups here, 1A, 1B, 1D, for example, the lines are a certain color, and then between 2A, 2B, and 2D, they're all the same color. And then in this case, there aren't actually any interactions across homology groups, but those would have been a different color as well. So it's relatively easy to change that script if you want to be able to do those things. Right, so circular interact.r, that uses the R-Circos library. And I didn't put the example. I mean, one thing you can do is just generate a random matrix of p-values, essentially, which in theory is showing you the p-value between all pairs of markers, and you can plot that. So that's what that line of code does, and then produces a file from it. I don't think that's the same picture that's in there, actually. That's funny. I don't know why I did that. Yeah, I put a different picture in there. So that image, didn't show anything significant. Maybe that's why I left it out. Weird. Okay. It should produce something. I think I picked a threshold there where it should produce some lines on the plot if you run that. Okay, so that's the visualization. I can go back to the selective phenotyping if people want, or if you have questions, then I'd say ask them now. Okay, I'll talk about this for the next 10, 15 minutes then. So selective phenotyping is something that came up for us because we had these populations. We had about 1,500 lines in a 4-way population and about 3,000 in an 8-way, and that's not really a number that's feasible to plant out in every trial and phenotype, and it would just be very expensive. So these days, phenotyping is really overtaking genotyping in terms of cost. Partly because you have to do it many times. You only have to do the genotyping once if you're just using one platform. You're doing it at multiple scales, in multiple environments, multiple traits, and so on. And so the question came up, well, how do we best choose which individuals to phenotype? Given that we have all of this genotype information on our individuals, can we do a better job than just randomly picking plants? And so the goal was to come up with a selection method which was general, flexible, and robust. So that meaning it could deal with missing data, it could deal with different types of designs, and that it was easy to do, basically, and that best captured the genetic information in the population. So we try and use the genetic information to inform which lines we're going to select for phenotyping. And the idea there is to maximize the genetic diversity. So if you have lines which are basically the same, there's no point in choosing both of them. So we're trying to avoid genetic duplication. And basically the previous options that were available were focused on very specific designs, F2s or backcrosses, for example. They couldn't handle missing data, and so they didn't work for our situation. What we came up with is called SP-Clust, and that's the name of the library as well, which may or may not be installed on your computer. That's why I wasn't sure whether I should talk about this part. And it takes a very simple approach. First, it computes the pairwise distances between all individuals based on how many alleles they're inheriting identical by descent from the parents. So this is designed for experimental populations. Those, again, are based on the founder probability. So it's taking into account the fact you may not be totally certain which parent it comes from, but that all gets factored into those distances between the individuals. We then cluster the distances so that we end up with k groups of individuals. So if you wanted to select, say, 200 lines out of a population of 1,000, your k is 200. and you select a representative from each cluster. So you're essentially trying to partition the space of diversity and pick lines which are spreading across that whole space. This is an image that you can produce with the package where essentially these red boxes are indicating each of those clusters, and then the blue stars, which are hard to see, are indicating the representative that's picked out of each cluster. And that representative is picked to be the most genetically similar to everything else in the cluster. You can also do it in multiple stages. So after running it once, it's possible, because this focuses on maximizing overall genetic diversity, it's not necessarily going to give you more power to detect a QTL. What you can do, though, is once you know where a QTL is, or if you've done some phenotyping before and you want to make sure to include those lines, you can run it a second time to refine the QTL position. So basically, you're going from genome-wide level to a candidate gene level, and then you can try and maximize the genetic diversity within selected regions of the genome. So they'll have higher genetic diversity, they're more likely to have recombinations. That gives you more precision in resolving your QTL location. That says basically what I just said. So when we do this, the way we look at how well we're performing, we looked at it both in terms of diversity, which it does increase, and in terms of how well we can localize QTL, width of the QTL support intervals. Down here, each of the box plots is labeled. So SB+1 is doing a single stage of this selection, and that was based on 200 individuals. SRS 350 is for random sampling with 350 individuals. So even with one stage of selection, you end up, because you have more diversity, narrowing the width of your support interval to something equivalent to 350 individuals. If you do it in 2 stages, again with 200 individuals, so 100 in the first stage and 100 in the second, you then get something which is essentially equivalent to random sampling with 650 individuals. So I have to reiterate, you're not increasing your power here, but you are increasing the precision with which you can localize your QTL. On the right is actually looking at the number of recombinations you're getting. So again, we have SP+1, so one stage with 200 individuals, SP+2. This third one, SP+2MR, is actually trying to maximize the recombination in the QTL region that you're looking at. And then SRS 200 is just random sampling with 200. You can see that with one stage of selection, you're not actually increasing the number of recombinations in the QTL region because that's not what you're selecting on. You're trying to increase genetic diversity overall. With 2 stages, you are increasing it, and obviously when you try and optimize the number of recombinations, you're increasing it even further. The implementation of this is an R package called SBclust, which is not on CRAN at the moment, but there's an archived version. And it should be back on there shortly. There are functions in there to compute the distance, to select the lines, and to visualize it. And that can be used for NAM populations, MAGIC, F2, backcross, essentially most of the populations you would get out of RQTL or NPMAP. It's a very simple sort of package. There's actually only, basically there's one function you want to use in it, which is called SPclust, and all you need to input is your data in a COS or MPCOS format, the number of lines you want to select, and the type of clustering method you want to use. And that's all that goes into it. And then you get plots out like this, and you also get a list of lines that have been selected. If you want to do 2-stage, you essentially put in the lines that you picked out of the first stage, and that's what informs what goes into the second stage. So that's all the slides I have in terms of topics. Is there anything else people have questions about, or that I've talked about, or that I haven't talked about? Yeah?
Previously, you talked about that circle and toward the interaction,
Yeah.
interaction. Do you mean like also the p-value is based on like QTL?
Yeah, so the p-value can be based on whatever type of analysis you've done. So basically that, in terms of visualization, that's just saying, okay, anytime I want to draw a link between 2 points on the genome, then I just want a matrix which is indicating when to draw a link. So I said for interactions, that's the obvious thing to use it for, but there could be other reasons you wanted to draw links between regions of the genome. The p-values you could get out of, so for example, if you wanted to do it with a biparental cross, from RQTL, if you run scan 2, I think you can get the whole matrix of p-values out. There's probably a way to at least trick it to do that. There's not a function in MP-map to do this at the moment, but I mentioned with epistasis, there's a couple different association type packages which you can run to get a full set of p-values across the genome. And essentially, you just need a matrix which has a p-value between any 2 points in the genome on it, and it'll draw a link when it's above or below a certain threshold. But yeah. Good.
For the founder probability, so like your program, anything like we can kind of interact with the parcel or the land population.
Mm-hmm.
I think it's very interesting to find out an individual line how much contribution from what area.
Mm-hmm.
So
So as far as the founder probabilities go, for biparental populations and NAM even, it's not such an issue because you're only looking at markers which are polymorphic between the 2 parents. So as long as they have 2 different values, you can always tell which parent it came from, right? If you have inbred lines, it's fairly easy to identify. If you want to phase those because you have heterozygotes, then you can calculate those founder probabilities as well in RQTL. And the way I'd recommend doing it for a NAM population is essentially doing it separately for each of those biparental populations and then putting that information together.
I have a question.
Okay.
It doesn't involve everybody. So, yeah.
Okay?
When you're clustering back when you do that, you come up with an error that says name attributes must be the same line as the vector, and it doesn't not need clustering. It just like cancels the function.
Okay. I might have to come look at it and see. Yeah. Yeah. If there's specific functions people have that have been working through the exercise, I'm happy to come around. So maybe that's the best thing to do at this point. Okay.